let's start out talking about the microtubules. It is one of the three components to the cytoskeleton. And microtubules are going to have some specific structure and function that goes with them. So they are the only one of the three that actually has a hollow tubular structure to them, as the name would imply. And basically what this does is it creates a structure that is very rigid. Well, I mean, it's still going to bend a little bit from side to side, but it's the hollow structure is going to make sure that you can't crush it very easily. And so if you think about a paper towel tube, for example, those things are pretty easy to bend side to side if you want, and you could damage it that way. But if you went on the two ends of a paper towel tube and tried to crush it, it would certainly fight against that pretty nicely. So microtubules in the cell are essentially the same way. They are then there largely for the as an internal skeleton support network, and are going to be located in the cell in such a way that it prevents the cell from being crushed. The organization and transport mechanisms that we talked about would also involve the cytoskeleton quite a bit. We'll talk about that here in a second. I'll show you how that works. And any cells that have cilia or flagella, those are actually going to be a modification of the microtubule network inside, and actually will be driven by the microtubules of the cilia or the flagella structure. The mitotic spindle, which we've also mentioned, is going to be a reorganization of the microtubule portion of the cytoskeleton. The other portions of the cytoskeleton are doing something else during the cell division process. So these would be somewhat special functions of microtubules. These would be what microtubules are doing for most cells all the time. The location of the microtubules is basically interspersed throughout the cell, and you'll find them somewhat collected near the nucleus towards the center, and then what they'll do is they'll radiate out towards the surface. The structure that actually controls what the microtubules are doing, maintains them, helps them grow, and is going to essentially also be used to reorganize them whenever that's needed, is what's called centrosome. And I just put this in here so that if you've heard about these before in a high school class, for example, centrioles are the animal version of centrosomes. We're going to use the word centrosomes just because some species or organisms don't have centrioles like we do. The centrosome is located near the nucleus, and you'll have sort of part of it that's near the nucleus, the other one essentially roams around, and both of those working together are going to help organize and maintain those microtubules as they radiate out. So let's take a look at what they look like here. So this is a micrograph, so an electron micrograph using fluorescence, where the microtubules have been stained green, and what you can see is how tightly collected they are around the nucleus, so you can really get a feel for how crowded the inside of a cell is. This nucleus is not going to be able to drift back and forth. It is essentially isolated in the center of the cell for protection, and just want to keep it safe and located where it is. And so you would have these microtubules crisscrossing across the nucleus here, and you can see how they radiate out towards the surface. So this would be where you'd find the plasma membrane, and these microtubules are radiating out towards the surface. So you can kind of imagine if somebody were to come in from this direction and push up on this particular side of the cell, then these microtubules are going to fight that pressure and protect the cell from being crushed. The spaces here would be where you'd find other organelles and components like the ribosome and other things. So this would be where the cytosol is found, and other components would be able to move around. You can also see fairly nicely the idea of a highway network. So if I want to move something from one end of a cell to another, these microtubules make a really good pathway on which to move things from side to side. The structure involves a protein called tubulin, and there's basically two different kinds of tubulin that go into microtubules. And what this does is it actually makes the tubule. Let me sort of just show you a little bit about what I mean here. So basically what happens is, and we'll just call them A and B, I think typically they go with alpha and beta, and what will happen is these alpha and beta subunits basically are going to form a hollow tube. And of course these will go all the way back, and I won't draw all of them, but there's going to be, you'll have B subunit this direction, you'll have A subunit this direction, and so what this does is it creates a polarity to the fiber, which is going to be fairly important, okay? And so one end is always going to be on the outside, one end will always be on the inside. I'm going to skin this back over here for a second. So when these things assemble, you'll end up having basically the way the subunit goes in this spiral is going to create a tube where one end is different than the other. So one end outside, one end towards the center. So of course the center one is the one that the centrosome is working with, and so at that end you kind of, if you want to shorten the microtubule, you'll take subunits off. If you want to lengthen the microtubule, you add them on, and it kind of extends out almost like you're sending a fishing line out. And that length would be controlled by the centrosome, and then of course locomotion, well not so much motion, but movement inside the cell is going to be controlled by motor proteins. So in this case what we've got is an illustration of these motions. So if you've got some kind of a molecule, and it's whatever they've stained dark here, if you have kinesin active, then kinesin is going to move things along a microtubule to the outside. So basically these motor proteins are going to work in one direction and one direction only. So if you want something to move the outside from the nucleus, you'll use kinesin to move you out. And if it's time to move it back in towards the nucleus, then the motor protein is going to be dynein. So these are two basically opposite, and in fact they actually are connected to each other, so that if we need to move out, this one walks along the microtubule, and then if we need to move in, this one walks along the microtubule and moves us back. So these two motor proteins work together in tandem to move things back and forth. So here would be some illustrations of things that maybe get moved around, and you kind of see the structure there. And that we call the plus end is out to the periphery, and the minus end would be near the nucleus or the centrosome. Of course the nucleus would be around here somewhere as well. So things like pigments, compartments that have things in them, lysosomes, endoplasmic reticulum components we can move back and forth. We just made something we need to secrete, we take it to the surface. Mitochondria might need to be moved to different locations in the cell. And so you can very much see how this highway system of these microtubules is going to play a role in how things move around the cell. So not only do they prevent the cell from being crushed, but they provide a really neat system for moving things around and being able to get things exactly where you need them fairly quickly and be able to use them at those locations. Okay, so next time we'll think about the intermediate filaments.